Chagos Archipelago is an incredible place. This remote group of tiny coral islands is surrounded by the cleanest seawater tested anywhere in the world, cleaner even than Antarctica. It's home to the world's largest living coral atoll. Coral reefs are the rainforests of the sea, an important breeding ground for a large number of significant species. Wildlife thrives here. Chagos hosts an abundance of fish that is 6 times greater than anywhere else in the Indian Ocean. It boasts the world's largest strictly no-take marine reserve, protecting an area twice the size of Great Britain from fishing and other extractive activities. Fish here have no reason to fear people. We work with leading organisations including the Zoological Society of London, the Royal Society for the Protection of Birds and the Royal Botanic Gardens Kew to protect the Chagos and its unique environment. CCT members lead research expeditions involving scientists from all around the world. Innovative techniques trialled here can inform the management of other large marine reserves. Each voyage makes new discoveries about the diverse habitats and teeming wildlife here. CCT scientists have monitored coral in the Chagos for over 35 years. Their research shows that these reefs have rebounded from climate change impacts particularly well. Further studies could help protect reefs worldwide. Because the Chagos Islands have no resident population, they have saved many of the stresses caused by human presence that reefs elsewhere suffer. The Chagos Archipelago is as important as the Galapagos or the Great Barrier Reef, a time capsule showing us how marine ecosystems could have operated hundreds of years ago. In a world with very few unspoiled wild places, we need to protect this truly precious and unique environment.
So good evening, everyone, to those of you here at the impressive Zoological Society of London and those of you joining us online. My name, if you don't know, is Sarah Puntan-Galea, and I'm the director of the Chagos Conservation Trust, and a very warm welcome to the second part of our 2025 annual general meeting, our speaker event. I thought it was important to start with this fantastic video created by the very talented John Slayer, who I hope is with us tonight. His images adorn our social media posts and our website, Chagos News. So thank you, John. But because the video reminds us what the Trust does to protect, conserve and educate the world about the beauty and importance of the archipelago and its precious wildlife and environment, something which is more important now than ever. So this is what our 6 speakers will be telling you about tonight. Each speaker will talk for just 10 minutes on a Chagos-related research subject before a question and answer session, where questions from the floor must be directed to a specific speaker on what they have spoken about today. And for those of you online, you will be able to send a question via the Q&A function at the bottom of your Zoom screen, and I'll see it here. So if you have a question for a speaker on a different topic or for myself or the board, keep it for the reception afterwards because we've only got 90 minutes. So without further ado, let me introduce you to our very first speaker, Dr. Pete Carr, currently our key rewilding project, Healthy Islands, Healthy Reefs Programme Manager, and an ex-Royal Marine Commando who worked on Diego Garcia for 5 years, 2 as a British Executive Officer and then 3 as a contractor's Environmental Director. Now, Pete's an avid birder. His PhD focused on seabirds using marine protected areas and is the author of 2 editions of Birds of the Chagos, books published by the Trust. And to quote Pete, for seabird islands to be restored successfully in the Chagos Archipelago, 3 essential ingredients are needed: rat eradication, vegetation management, and time, which is a perfect starting point for his good news talk to start us off this evening, which reports on the success of a rat eradication project conducted in the south of the Chagos Pyrospanos Atoll. Vache Marine, 10 years on. Pete.
What? Wow, that was a great introduction, wasn't it? Thanks, Sarah. I'll let you into a couple of secrets before I start. The first one is I'm really suffering from imposter syndrome at the moment because I've met my fellow speakers and I know some of them, and I've had a sneak preview at some of their talks, and you're in for an outstanding evening. And I really feel like I'm just the cheap warm-up act at the moment. And the other thing, I must make a confession. For those of you who know me, everything I do, I plan down to the finest detail, and that's probably something to do with that background as a commando. But Vache Marine 10 years on all came about by happenstance. We didn't plan to go back 10 years on. I was working with a phenomenal PhD student called Hayley McLennan in October, November last year, which coincided for 10 years since we did the project. And we had a really great senior fishery protection officer on board with us, a guy called Daniel. And between the two of them, they worked it that I ended up on Vache just about to the day 10 years on. And I can't thank them enough for that. Moving on to the talk then. You said I had to wave it over here. There you go. Okay, so here's what I'm going to be talking about. First of all, I'll just briefly talk about the location of Vache, why we chose Vache. I'll talk a little bit about the eradication operation. It really was an all-hands-on-deck operation. And then I'm going to do a little video. And again, happenstance, we got on to Vache Marine. Hayley McLennan got out her phone and says, "Hey, Pete, do you want to make a little video about this?" So it's not John Slayer standard stuff, I warn you that now. But it will give you a flavour for those of you who haven't had the opportunity to go to Chagos to see a little bit of what the island's like. And then I'm going to conclude with just some thoughts about Vache Marine 10 years on. And then there won't be questions, because questions are going to be at the end. So first of all, where is Vache Marine? This is a paper we published in the Chagos News a couple of years ago. They're colour-coded islands. Orange is obviously rat-infested. Green are rat-free. And then there's one island, if you have a look at Pyrospanos, down in the south. I'm going to have to shout and show you this one. But that purple one, called Vache Marine, you may be able to see that at the bottom. That's Vache Marine, and that's the only island in the Chagos so far that's been successfully eradicated. I'll just give you a little bit of history about here, and if anyone was at the AGM, you may have heard me say that I just think I'm the keeper of the person driving the rewilding at the moment. And I say that because all of this healthy islands, healthy reef stuff, in my opinion, started way before me, way before even the Chagos Conservation Trust. There were some remarkable people working out in the Chagos in the 1970s, David Bellamy and the much missed Dr. Charles Shepherd, Professor Charles Shepherd even. And they wrote a paper, they wrote a letter to Nature in about 1974 saying, hey, there's something really wrong out here. And it was rats. And they noticed even then that seabirds and rats weren't getting on. And as a result of their paper in 2006, there was the first attempt at rat eradication in the Chagos on Eagle Island. And for those of you who know, excuse me, any of the characters that were involved in that, it was a real Herculean effort of what they did and how they survived. Literally how they survived is just fantastic. Unfortunately, that was failure. So there we ended up that there was a little bit of faith lost in can we do operations in the Chagos. And in about 2011, the Chagos Conservation Trust, which I was a trustee of around about that time, we decided to try again, but we decided to try again at a much, much smaller scale. So Vache Marine is 15 hectare. Eagle Island that those chaps tried, or that team tried, is 252 hectare. Anyway, so Peros Banos atoll in the middle, Vache Marine is that purple island round the bottom. And just before I get into the meat of the talk, the Healthy Islands, Healthy Reefs, our application that we have in at the moment will be to clear those 4 islands up the top, the north of Peros Banos, which will produce an uninterrupted chain all the way from Pas round to Vache Marine. That is the intent. Okay, so why did we choose Vache Marine? The most important thing about just about any eradication operation, but certainly with rats, is that you don't want them to reinfest the island, reinvade the island once you've successfully removed them. Vache is surrounded by deep water and it's about 4 kilometres away from any other island. And theoretically, rats shouldn't be able to cross that. So if we eradicate them, no rats can get across from the islands like Coin, which is 4K over. It's also a small size, as I said. The first attempt that they made out there in 2006 was 252 hectare. That's just incredible. So we chose Vache because it was 15 hectare. And it was readily accessible. It's not the easiest island to get on, but it's by no means the hardest island to get on. And the good thing about Vache is that it was never heavily farmed for coconuts. There was a little bit of coconut exploitation going on there, but there was still quite a lot of native vegetation present. And also Vache is in what is now the Eastern Peros Important Bird and Biodiversity Area. For seabirds to recolonise, proximity to that island is important. So Vache is dumped right in the middle of an IBA. Theoretically, once rats are eradicated, seabirds can make their way back. Okay, so the eradication operation itself was a real feat of all hands on deck. It was a real pleasure to orchestrate it all, and it started off with the Bight administration at that time giving us permission to use what was the Bight patrol vessel, the much-loved Pacific Marlin, and we had all our stores flown into Diego Garcia, all the bait, all the traps. It was loaded up onto the Bight patrol vessel, and then we had the military come with us, and it was the military, along with the MRAG Senior Fishery Protection Officer, myself, and a rat technician from New Zealand, who did the work on Vache. On the way up there, we conducted training, as you can see in that middle slide, where all those really interested military folk were taught how to handle bait without poisoning themselves. We then used the rigid raided craft, which is that far right hand where I'm looking, to get the bait ashore. Unfortunately, at the day we chose, the weather was a bit gnarly, and we had some fairly hard times getting all the bait ashore. We then, once all the bait was ashore, we had to cut lines. When you're doing a hand-baited trap operation, you have to have a trap in every 5 meters squared of the island, so we had to cut lines all the way through the island, criss-cross, which took us several days. And then we started baiting the traps. And we then left the island for 14 days, and then we went back and repeated the baiting of the traps. And then that was that operation complete. And on a tropical island rat eradication, you have to wait 2 years before you can declare it a success. For various different reasons, we didn't get back in 2 years. We got back in 3 years. We had an independent person verifying that rats had been eradicated, and in 2017, Vache Marine was done in 2014, it was declared a success. And then 10 years later, from 2014, when we conducted it, I ended up back on the island. And I did say that Hayley, when we got on the island, that's Hayley on the left there, she said, "Hey Pete, why don't I get my iPhone out?" And Izzy's laughing because she knows she's in this video. Why don't I get my phone out and make a video about this? So I'll tell you again, it's not John Slayer standard, but for those of you who haven't been out there, you'll get to see what the island's like and you'll get to hear the results. And I'm going to stay within my 10-minute allocation.
Research in the Chagos Archipelago has revealed the key role that seabirds play in island ecosystems. By foraging offshore and returning to deposit their guano on land, they transfer otherwise inaccessible oceanic nutrients onto islands, fertilizing plants and nearby coral reefs. However, islands around the world are being degraded by the introduction of invasive rats, which predate seabird nests and decimate their populations. Disrupting these critical ecological trends has devastating impacts for biodiversity, island resilience, and carbon capture. In 2014, Vache Marine became the first island in the Chagos to have rats successfully eradicated, and 10 years later it remains the only one. Carr, who led the original rat eradication on the island, has returned to monitor the success of the project.
Well, we've just landed on Vache Marine, and it's just about 10 years to the day when the Chagos Conservation Trust conducted a successful eradication of rats on this island. So I'm back here today to see if the biodiversity has improved and to double check that there are no rats present. You see the really young chicks in the middle. Certainly more white terns on the island than previously.
Sea turtle nests are also predated by rats, so a rat-free island would be good news for the turtles that lay eggs here too. That's always a good sign, you know, because rats take seeds from the floor. There's one of the old bait boxes that got left behind by the looks of it. The eradication was successful. You wouldn't get this amount of seeds sprouting on an island with rats. That's a good thing. That's a rat, but very old. Very old. So that concludes our look round Vache Marine 10 years on. What have we found? Well, there's certainly been an increase in the small finch-like bird that we see here, the red-foddy. Their numbers have gone up probably 100%. As to the other stuff, there's none of the exciting birds that we hope to find, like bridal terns or frigate birds. But the common birds, the rat non-sensitive birds like white tern and common noddy, their numbers have slightly increased. But the most important thing is, after we've looked round the interior, there's lots of seeds coming up inside in the forest, and that's a good sign that rats were eradicated and there are no rats left on Vache Marine now. So let's let time do its thing.
So,
I've done a lot of thinking about the after effects of Vache marine. I really have. And first, it's obvious to me, looking at Vache marine now, that and we know this and we proved it with some of the Bertarelli programme science, is that rewilding an island is a two-phased operation. One of the problems with Vache is that the habitat isn't really conducive to breeding seabirds. So had we combined rat eradication with some hardcore vegetation management, there may be a slightly different result. And we could have enhanced that, I think, if we had used bird attractants like calling from colonies, or even using decoys, which have been used around the world. But they're just two slight things, in my opinion. What rewilding really wants on these tropical islands is time. It takes time for birds to find, it takes time for birds to find the island, and then it takes time for them to understand that it's a safe place to nest. And 10 years in rewilding isn't long enough, certainly on islands like Vache, where the habitat isn't exactly what seabirds want. I would like to thank an awful lot of people, but some of the people up there are the real stars, and particularly prior to us conducting the rat eradication operation out there, I had two Chagosian environmental students who worked with me in 2 different years, gathering baseline data, and they were absolute stars. And Colin Clubb and Grant Harper also deserve mention in dispatches. There you go. Thank you very much indeed.
Thanks, Pete. It's so heartening to have some pre-project good news as we wait for funding to get started on our Healthy Islands, Healthy Reefs project. And if you want to know more about our Healthy Islands, Healthy Reefs project, there's lots of information on our website, and Pete also wrote 2 articles, one in 2024, one in 2023, which is still up to date. So, now, I know many people are here tonight and joining us online from the US to hear the next speaker and author of this year's Chagos News cover story about the infamous Chagos coconut crab, which holds the Guinness World Record, and I quote, Earth's largest land-based crustacean with up to a 40-inch claw span and a 10-pound body. Impressive. So Dr. Mark Laidre is a professor at Dartmouth College in the US. He's been researching coconut crabs for nearly a decade after encountering them on the Chagos Islands, but his work at the scientific interface of behaviour, ecology, evolution and conservation goes much further back than that, and he's been researching the shell housing markets of marine and terrestrial hermit crabs for 2 decades, co-authoring more than 50 scientific publications. His current research project is funded by National Geographic and focuses on coconut crabs across the Indo-Pacific. So please tell us more about coconut crab conservation in the Chagos and beyond, Mark.
I just want to contradict Pete for a moment and say that I'm very honored to go after you. I think all of your contributions to conservation are incredibly admirable. Let me just see if I can do as well as you in using this remote. The other thing I'd like to say very briefly is that I think ants are a fascinating creature, and I hope you will enjoy a later talk, which is by another invertebrate. Okay, so I would like to tell you about coconut crabs today. As one of the few Americans here, I will very briefly begin by saying, I'm sorry. And I really am. Democracy, you don't always get who you vote for. I'm sure, as you may be aware, there are a lot of challenges with this right now. I'm going to put politics aside and talk about what I do know, biology, but I'd like to mention one more political thing, and that is a criticism of the UK, a brief one, a small one. How could you ever take Charles Darwin off your money. I do not even understand that. Please explain it to me in the question and answer period. And I say that for many reasons, quite seriously. Like many biologists, Darwin is one of my biggest heroes. He was also one of the early contributors to our understanding of coconut crabs. If you read Voyage of the Beagle, some of his observations really went into great depth. 2019, I was here for a CCT talk, and you would have seen many Darwin quotes, about a dozen of those throughout the talk. But I will have just one to begin, which is that Darwin, like many people, were quite infatuated with the size of these animals. Quote, grows to a monstrous size. Well, one thing that Darwin didn't highlight, and which is, I think, quite important, is that the way this actually happens is life history. And life history for these coconut crabs involves going through a shell-dwelling stage, like the terrestrial hermit crabs that are their closest evolutionary relatives. And you may be aware that in terms of the value of these shells, these are quite valuable to hermit crabs, but they were actually the very first currency of humans, shells, cowrie shells in particular. And when you think about these shells within that context of their value, it becomes very interesting because like many forms of so-called animal architecture, and shells are most certainly an example of that, these are in effect a housing market in nature. And this includes many things that I won't talk about in this talk, but all sorts of really wonderful forms of built structures and modifications of the environment that animals make. Now, shells have been of particular interest to us. For well over 20 years now, we have been studying those. These are a selection of papers from our work at our long-term study site in Osa Peninsula, Costa Rica, where we work in particular on this very interesting phenomena, which is that crabs also remodel shells. So on the top right, you can see an architecturally carved out shell. These are from Cenobita compressus, the species that we study in Costa Rica. And these architecturally remodeled shells are something they've actually become so specialized on that they cannot live after a certain life stage in the original unremodeled shells built by snails. So this has really favored a highly, highly social species. But a lot of this work also informs things that we may think about with respect to other crabs. Very briefly, I just want to highlight the work of this incredible scientist whose name was Jocelyn Crane, and who wrote a book entitled The Fiddler Crabs of the World. And I'm right now working on a book for that same press. That is my intention to publish with Princeton Press on Nature's housing market. But I just want to note something. Her book was 766 pages. So I do think, and I think sometimes in this current culture, we're obsessed with these little bits of information, tweets and social media, but that to me is what a real scientific contribution is. And I don't intend for a 766-page book, but that is quite admirable. So for a decade, I have been teaching classes. I like teaching a lot. Teaching is fun. I've created many, many classes at Dartmouth. Teaching, though, is not my favorite thing. I love research. I love being in the field, and time is the most essential resource. I was made well aware of that by this person, who was my grandpa and who taught me, I think, all the most valuable lessons of life, including how you get a coconut down from a tree, which he was doing that in his mid-90s. We're very excited to have gotten a really huge grant from National Geographic. This will fund our work for the next 5 years, and this is what I intend to be my focus for the next 4 to 5 years. I want to give you kind of a brief overview of that. Apologies, Chagos, you're not up there, but you'll be there in a moment. We're centering this work on Palmyra Atoll, which is run by the Nature Conservancy. And the reason for that, quite simply, is that those islands serve as a natural laboratory for experiments. And one of the core goals of this long-term project is to create whole island experiments in which we will manipulate the housing market, changing shell availability on control versus experimental islands, and looking at how that impacts ultimate recruitment of adult coconut crabs into the population, because these animals have to navigate through a housing market that not only is saturated with other members of their species, but with 3 other sympatric terrestrial hermit crabs that are permanent members of that housing market. On top of those whole island experiments, our goal is, and hopefully some of you folks know these incredible scientists, Peter and Rosemary Grant. Modeled after them, they have tracked unique individuals on islands for decades. And those were Darwin's finches. Our goal is to track unique coconut crabs. And so we will be tagging, and the goal really is this, every individual on all the islands we study. That will be thousands of individuals. We have 20,000 RFID tags ready to go. Our point in doing that is this will allow us to track the molting and growth of unique individuals across time. Not only that, but I like to have ambitions that are far beyond even kind of short-term 5-year grant projects. My hope is to look at evolution on these islands in terms of what crabs are still there, which ones survive, which ones have higher versus lower reproductive success. And these unique tags give us that gateway into that. And it's only when you have individuals uniquely tagged that you can do that. A third element of this project, and these are kind of the 3 core elements, is GPS tagging, which we're doing with our colleagues at the Max Planck's Institute, which I'll visit after this visit to the UK here, is we're GPS tagging adults, and we want to know where they move around those islands, where they release their eggs, and in detail what their spatial use of that island is. Naturally, you try to moonlight a lot of other things on it. One thing I'm quite intrigued to do is to think about how these housing markets are carved up. So up there you have 3 of the core species that live sympatrically with coconut crabs. These are Cenobita brevimanus, rugosus, and perlatus. One of my colleagues at Dartmouth that I respect the most, Mark McPeak, he's an amazing theoretician as well as an empiricist, but he wrote a wonderful book about coexistence in ecology. How is it that all of these different species coexist? Right now we don't know that, but these are experiments we want to do on the fine scale to think about how these different individuals utilize perhaps different shell species and how some of them are managing to still coexist despite this extreme competition within the housing market. We also intend to put out a network of camera traps, and this kind of has a number of things. One thing we want to look at is what are coconut crabs doing out there? Turns out sometimes they take large birds. I'm sorry, Pete, for that. Turns out they do that on other islands. So someone named Jennifer Apu out in Seychelles had observed a very interesting case of one going after a crow. And I think of crows as very intelligent birds. Red-footed boobies, I'm not so sure, but crows are most certainly very intelligent. What we want to do with these camera traps is center them around burrows and have a network such that we can see all of the things that these animals drag back into their burrow, as well as all the very interesting, this is kind of my own core interest, is all the various social interactions, territorial interactions, and mating interactions that take place at the epicenter of these burrows. Coconut crabs aren't doing great, like a lot of things out there. Our hope with a lot of this work, even though I view myself as kind of a fundamental scientist, is I hope some of it can ultimately be applied in other locations, not only Chagos, but all over the world. And one of our goals in particular is thinking about working with other scientists and human beings. It turns out, like hermit crabs, which do cooperate, smaller and medium-sized ones cooperate to work together to evict larger ones, bad ones that are kind of occupying the top of the housing market, just kidding. So scientists too need to cooperate such that they can kind of do the best. And so something I'm very excited about, and I certainly hope this Darwin Award is funded for the Chagos Archipelago, because if it is, I would very much be happy to devote resources and people to tagging obsessively all of the coconut crabs on these islands that are being eradicated of rats. And the reason for that is I think this relationship is quite complex. I kind of highlighted a few of those elements in that Chagos News thing. We don't know all the ways that rats and crabs interact or their complex relationship. One thing that is clear is that across their life history, a coconut crab's relationship with a rat varies from being a competitor to a predator to prey. And all of the nonlinear ways in which that will play out when rats are eradicated, I'm not sure that we can really predict what that outcome will be. But whatever it is, we do need to know that. And knowing that on the individual level with uniquely RFID tagged crabs, I think is quite important. I'm looking forward to getting a copy of Pete's second edition of the bird book, and I mean that very genuinely. I have a signed copy of the first edition, so I hope to get my second edition signed. Something, though, that is of interest to me is, surprisingly, not infrequently, I get emails to say, what crab is this? And of course, it's not a coconut crab. That's relatively easy to identify. But there are a whole suite of dozens of crustaceans in Chagos, many of which people need a guide for. And I just want to call this out to CCT. I would be very happy to create some sort of little pamphlet guide, nothing kind of as wonderfully book length as Pete's bird guide, but some sort of pamphlet guide that could be helpful, I think, to not only researchers, but to kind of the lay persons who are interested of all of the different crustaceans that are out there on Chagos. I will note too, I was tickled to see this Christmas card that was drawn by Martina. Many years ago, we created our own. This was my collaborator, Sarah Smith, who's an artist, drew a very wonderful little coconut crab who, while it doesn't have a shell, it does have a very toasty, warm sweater with its wine glass. And personally, I'm very glad that these two Christmas cards did not meet, because I don't think that type of Christmas or red is necessary. But speaking of that, I do want to say, like, one of the reasons to come here is definitely to think about the connections with other projects that are going on. So, you know, if there are things that people are studying, whether they be corals, whether they be birds or other things, please kind of tell me how we can help, or what interesting intersections you can see. Because I think not just being too taxa specific, but thinking about these kind of synergistic elements is very vital when you're working in a lot of these remote places. I will end by saying that way back in 2019, when I came here for a CCT plenary talk, I donated a coconut crab conservation shirt. I'm wearing one of these to CCT, and I was told I would get in return a CCT t-shirt, and apparently, and this is really why I came all the way here to the United Kingdom, is to get my CCT t-shirt. I just want to end by noting that this is our dog, Darwin. As you can see, she's not merely an emotional support animal, she's an intellectual support animal. She's very well trained to answer emails. If you don't get an email reply from her immediately, just know that Darwin is on it, and she's still figuring some of those things out, but she's very good at that. I will end there, and thank you very much.
Firstly, patience. We may have gifts for you later. But I think it's very important points you made, that being a very small trust, we need to work together to get more done. So thanks for that. We'll definitely be calling you about those leaflets. And if you want to read Mark's Chagos News article on this iconic species, it's available on our website and January's Chagos News, and also in the British Library archives. Now, from one Dr. Mark to another, sitting next to each other, Dr. Mark Spalding needs no introduction, being the current British Indian Overseas Territories Administration's Chief Scientific Advisor and a great friend of the Trust. He first visited the archipelago as a PhD student on former CCT Chair Charles Shepherd's 1996 expedition, and keeps returning, working with the then Friends of the Chagos and Biota. Alongside his Chagos work, Mark is a senior marine scientist with the Nature Conservancy and an honorary research fellow at the University of Cambridge. He has spent much of his career looking at the global distribution and status of marine ecosystems, publishing the World Atlas of Coral Reefs back in 2001 and the World Mangrove Atlas in 2010. With 8 other books and over 130 publications, Mark has a pretty broad understanding of the world of coral reefs and conservation. So his description of Chagos as the world's most important coral reef wilderness has some considerable authority behind it. But tonight, he's going to tell us about coastal lagoons in his talk, Terns, Turtles, Salt Marsh and Sharks, Barachois, a unique habitat in the Indian Ocean. Mark.
Thank you very much. So this is possibly a bit of a ramble, and I'm kind of throwing down a gauntlet. I think everyone who's talked so far, and everyone who does talk about Chagos, talks about what we don't know, about how much more there is to learn. And so I'm going to do that, I think, in spades, really. Almost the same picture that Mark had, but I had my clouds are in a different place, and that's really important. You all know what coral atolls are, these circles of coral with a deeper lagoon in the middle. In fact, Darwin was inspired, we're going on about Darwin. Darwin was very much inspired to come up with his theory of the formation of coral atolls by the maps that he saw. He never went, but the maps he saw of Chagos. But what you see in this picture is these little inlets coming from the lagoon into the island of Diego Garcia. And the Chagosian people called these barachois. And my argument today is that we ought to be looking at these a lot more than we have been, because I think they're incredibly important. I first started thinking about these a few years ago. I go out every year in my role as a chief scientific advisor, and it's a funny mix of talking to all the sort of key people, but also just wandering about and trying to take it all in. And I went to see one of these tidal inlets, these barachois, and saw all of these little dots. All of these dots are fiddler crabs, and they're really, really beautiful, but very, very timid. So I thought I'd get up early one morning, and you can actually cycle to this one, and I cycled to this one, and I sat still. All the crabs had disappeared, but if you sit still long enough, they all start coming back out. And I'm sitting there and gradually getting closer and closer and seeing these tiny little crabs, but everyone has different coloring, and they're really beautiful. And this is a real Diego Garcia moment, because I'm sitting there in what felt like the wilderness, and suddenly, over the loudspeakers, which I couldn't hear, I hear and then suddenly the national anthem starts. And that's Diego Garcia for you. It's kind of this weird mix of nature and weirdness, I suppose. But anyway, so I started thinking more and more about these, and I've now sort of drawn their outlines on a map. But these little tidal inlets, so you've got the big lagoon, it's a big tidal system, it's a macrotidal system, and the tide flows in and out of these lagoons, and there's about 16 of them. The total area of them, and they're really all on Diego Garcia pretty much, is about 250 hectares, which is pretty big, same size as Eagle Island. So they are, if you covered those with, I looked this up, I've got the numbers in front of me. If you covered those in solar panels, you could probably power a small town. I mean, there's a large area, and they are, in a sense, solar panels. They're getting a heck of a lot of sunlight, but there's nutrient there, and there's moisture, so they are pretty interesting. And if you go to them, because the tide's flowing in and out, there's lots of fish that will pour in and out of these things. As the tide comes in, the fish all swimming in. And these are mullet, and they're feeding off the sort of algal scum and stuff on the surface. And you'll see many rays swimming in and out of these systems. In fact, the diversity is pretty exciting. Quite often, the inlet into these barachois is a narrow channel. When you get this concentration, and as the tide's going out, some fish are being swept out. You see the terns dive bombing to try and get the fish. Well, I'll talk more about the turtles. You see lots and lots of sharks, juvenile sharks, lemon sharks in particular, cruising up and down outside, and as the tide comes in, the sharks swim in. The rays, as you can see, the rays are going to be feeding on shellfish. So there's a lot of action around these places. And probably what's driving that is this productivity. So the top right is a grass called paspalum. It's, I think it's fair to call these patches of grass saltmarsh, pretty much like you'd have in the Thames estuary. It's possibly an invasive species, but it's found all around the tropics, paspalum. But in addition to that, you get these mossy algal types, and you get these blue-green or cyanobacteria, which I've sliced through. It feels like you're walking on sand, but actually if you take a slice through it with a penknife, you can see it's green and pink, and that's cyanobacteria. Same stuff that builds stromatolites. And in fact, there may even be stromatolites in Chagos, which would be pretty cool. We've got to find them still. But they are probably highly productive with this perfect setting, and there's a lot of wildlife behind them. And Holly's here, and I've grabbed some somewhere, I think. There she is. And she's come up with this fantastic paper, because there are lots and lots of turtles. In 1996, on Charles's expedition, we all went down to this place called Turtle Cove with Jean Mortimer, another famous turtle biologist, and got super excited because there was this narrow sort of small river-sized channel going into one of these huge lagoons, and there were turtles just popping their heads up everywhere. And Jean got super excited, and work on Turtle Cove has been going on since then. This is, I think, a drone photograph, isn't it, Holly? But every circle is a turtle. And through that work, through this combination of Mark Laidre capture, tagging, and drone surveys, she's mapped out the density and the movements of these turtles. So these different colors are 6 different turtles that were tagged and tracked. And from that paper, she's shown, in fact, that Chagos has the highest density of juvenile hawksbill turtles anywhere in the world. These things are just packed in Turtle Cove, this density of these young turtles, and they're basically coming in to feed. So that's what we kind of do know, as much as I'm sure there's more to know. Pete will tell us with great enthusiasm about the best places to see all the waders in Chagos, is again the barachois, and you'll see lots of different species and quite large numbers. And I suspect, but I don't know if we know this yet, Pete, that there's probably a higher density of waders in DG than most of the other atoll islands around the world, probably because of these barachois. Of course, the barachois are also of interest to people. The Chagosians used the barachois. I found most of the old names for the barachois that Chagosians used. Quite cleverly, 1 or 2 of the barachois, they built barriers on these narrow channels, so they were able to catch lots of fish very easily through that. That obviously, that's not happening anymore, but they're very popular, some of these shallow areas, either in the barachois or just offshore, for a very small number of people who do fly fishing. Interestingly, if you go to Alphonse Atoll in the Seychelles to go fly fishing, you'll pay hundreds or even thousands of dollars a night for the privilege of going fly fishing. And from what I understand, the fly fishing in Chagos is better. So it's a fairly niche sort of hobby, but if you want to do fly fishing for bonefish, permit and so on, these are fantastic places for that too. So how are they formed? And in fact, people have been interested in this barachois for a long time. So this is one of the papers by Bourne and Lancaster from 1888, describing the formation of barachois. David Stoddart, another giant of coral reef research, and Chagos, wrote this huge tome in the Atoll Research Bulletin, which also has a lot of information about his observations of barachois. I also put this from the Chagos News, this diagram by Charles Shepherd, the late great Charles Shepherd, which helps to explain what Bourne and others thought was going on. So if you look at many of the islands, and that's what these profiles are here in Chagos, they have a higher rim, and they drop down in the middle like a basin. Sometimes, there's no pointer here, but sometimes the top left one, they drop below sea level in the middle of these islands. Or they're certainly close to sea level. And so when you have a breach in that rim, the tide can flow in and out. So that was one of the theories, at least, for how the barachois are formed. And we've kind of got a bit of evidence of that. So the 2 photos on the left are barachois Lubin in southeast DG. And that's been there for a while, but you can see dead palm trees on the edge. It looks like it's probably expanding. And that's the road on the highest tides, and that's now flooding, I think, pretty much every month on the highest tides. And the channel's essentially being reformed, and that's becoming a barachois. The top right, I'm sorry, Pete, this is one of your restoration sites. In 2019, we had some really super high tides, and the sea flooded in to new areas that it hadn't flooded before. And it killed all of the trees that Pete had carefully planted in that restoration site. And a little further along, there was just some coconut chaos, which also got flooded. In fact, the restoration site's somewhat recovering, but the coconut grove there is now becoming, I think, a new barachois. Almost there. So I think I wanted to talk about the barachois in a sense to throw down the gauntlet. I think here we've got something that is really very special, but to find out more, we need to do a lot more science. There's almost nothing we know. The scientists going out there, they're doing the birds, the vegetation, or the coral reefs outside, but actually we've got these barachois. I've had a look on, I've been searching for some time on sort of world maps and through other papers. There's a couple of barachois, named called barachois in the Seychelles, but they're small and much less impressive. There's a couple of things which could be barachois in the Maldives, and there's a load in Kiribati in the Eastern Pacific. One thing they all have in common is they're outside of the range, the easy reach at least, of mangroves. In many other coral atolls, these things would be full of mangroves, and the mangrove trees would change the dynamics completely. But we don't have, there's a couple of mangroves in the north of Chagos, but they haven't reached DG, and I think that would make a huge difference. So you've got these huge, flat, intertidal solar panels, effectively, being washed by the tide on a daily basis. I think we need to know more about the productivity. We need to know about what species are there, and the numbers, and the biomass. Just on the crabs for Mark, there's the crabs everywhere, many different species. There are sort of zones of crabs. Also different species of rays coming in and out and feeding in these things. I put the manta ray in there deliberately because I suspect that these things are actually generating nutrients and food that's going way beyond the barachois and into the atoll and possibly beyond the reach of the lagoon of the atoll. And maybe we've got another story here that's not all that far from the nutrient story of the birds. And I'd just love someone to get out there and do it. It won't be me, but I'd love to help. Yeah, so that's it. That's about the barachois. Thank you.
Thank you, Mark. And if any of you haven't read Mark's most recent report on the state of Chagos coral, please grab him at reception and he'll tell you where to find it. And please do read it. Unfortunately, it makes for a bit of somber reading, but it's a stark reminder why everyone here is fighting so hard to protect and conserve Chagos coral and all the archipelago's nature. So that's a perfect segue into the next talk, which looks at the ever-present threat of illegal fishing in the waters surrounding the Chagos Islands. Sadly, Dr. Claire Collins, who's a postdoctoral researcher here at ZSL, as well as an honorary lecturer at Exeter University, had to pull out last minute, but her co-presenter and PhD student, Abishek, will now present the whole talk. Abishek is from India's Ashoka University, where he's studying the human dimension of illegal fishing in the Indian Ocean region. His research examines Indian policy framework to tackle illegal fishing, drivers of illegal fishing behaviours in fishers, and uses participatory mapping to track fishing patterns. So please tell us more.
Thank you, first of all, Chagos Conservation Trust for inviting me. It's a privilege to be a part of such esteemed speakers list. So today I will be talking about illegal fishing in Chagos, its dynamics and impacts on sharks. So basically I will be talking about three parts here. First is about role of Diego Garcia and for sharks. So this work is mainly done by Claire as well as Chris. They both are postdoctoral fellow in University of Exeter. The second part, I will be talking about the impact of illegal fishing, specifically on the Diego Garcia. And then I will be bringing on my study, which is the PhD study I'm looking in, and the aspect of illegal fishing, especially the drivers behind it. So first, the study which is conducted by Claire and Chris, they are using this instrument on the left, which is called Baited Remote Underwater Video System, which is BRUCE. So it's a GoPro which is put under a steel platform, and then there's a bait which has been put in the white kind of thing you see there. And they are being put in 75 different sites around Diego Garcia. And the idea was to understand the presence of sharks as well as what shark species are there in and around Diego Garcia. So this data is being collected in last November 2024 and currently is analyzed by Lauren, which is in the audience. So this is a video of highlights of 90 hours of data that has been collected. So I'll just play this video. So this is the site where it was put, and there were species like lemon sharks. So unfortunately, there's no audio here. Surprisingly, there were no silver-tip species which were found in any of the BRIV devices. However, there were a lot of grey reef shark species which has been recorded. Yeah. So the idea behind collecting this data is to understand if there is a nursery of shark in that particular area. And if that exists, this work will feed into important shark and ray areas work as well. So basically, the studies in past have shown that Diego Garcia might work as a refuge because illegal fishing is completely prohibited in the area due to military presence. And this has also been supported by one of the study done by Dunn et al. So here it shows that how in northern atolls, the grey reef and silver-tip species were almost none, but when it comes to Diego Garcia, the numbers were pretty high. So probably they are taking refuge in Diego Garcia, and this place can be a refuge for other species as well. However, illegal fishing in this particular area has been a problem, and this has been shown in Claire's previous study as well, where a lot of fishers from Sri Lanka is moving in this area. However, there is a spike from Indian fleets as well in this particular area. So this is where my PhD comes into play. So I'm doing my PhD from Ashoka University, and the idea where I am mainly focusing on is understanding the drivers of illegal fishing and the fishers' behavior. And these are mainly focus area of my PhD research. I am looking at the policy framework in India to tackle the illegal fishing, what are the drivers of illegal fishing, and in order to map the illegal fishing behavior, we are using a participatory mapping technique. And we are also looking at why, because a lot of areas in India is not using vessel tracking technologies, so why they are particularly not adopting those technologies. So this map here is from my pilot study done last year in June, July for a month. So basically, we conducted participatory mapping, and it clearly shows that people are moving in illegal territories as well, and this also includes Chagos as well. So why this illegal fishing is happening is what is our focus is, and I'm looking at why studies have shown that data for these, in order to map the fishing pattern, the data especially coming from vessel tracking platforms is not fully reliable, and the reason is mainly because a lot of vessels are not using these tools. The adoption is not as high as compared to western part of the world. And also a lot of fishers are manipulating with these tools when they go out in the illegal areas. And as well as studies have also shown that there is a poor quality transmission when it comes to South Asia. So this is where I'm trying to address this issue, is to how to, why they are manipulating, and if they are manipulating, how to monitor this movement if they are doing. And this is where I'm looking into how this can be a concern regarding to Chagos, as well as particularly sharks as well. And I am using this participatory mapping technique, which I will be talking about this in the later slides as well. So what we are doing is we are using physical maps and working with fishers, and we are trying to map the fishing areas where they are moving in. And we are particularly focusing on shark fisheries and how these vessels are interacting with particular shark populations, and if they are moving their fishing effort, if it is in high seas as well as in Chagos. We are particularly focused in Kanyakumari, which is the southernmost district of India. And why Kanyakumari? Because Kanyakumari has been in focus when it comes to illegal fishing, and especially illegal fishing in Chagos. And to be more specific, a particular region called Tuttur. So this village is known to be doing targeted shark fishing and moving in the areas of Chagos, and has been shown by previous studies as well. So this is where our study site's focus is. This is one of the map that we are using for participatory mapping. And these are some of the data points that we have collected from the pilot study done in June, July previous year. So this has also shown that fishes are moving in the waters of Chagos as well, and further to Chagos as well. These are some of the pictures from the village, focus groups conducted, interviews conducted as well. This picture is one of the participatory mapping exercise done. So currently, this data collection process is ongoing as I speak here. Moving on to the policy study that I have done as the first chapter of my PhD. So here we have tried to focus on the policy aspect, as policies plays a very critical role when it comes to tackling the illegal fishing through the official channel at least. So the issue in terms of South Asia regarding the illegal fishing regulations has been shown in by other studies that the implementation has been weak. And that's what we were looking in the context of India, that if our policies in order to tackle illegal fishing is strong enough, and comparing it with the FAO's International Plan of Action Against Illegal Fishing. And we were also looking at online evidence of their implementation. And this study is done, and some of the key findings that we have identified is that inconsistent vessel tracking has been one major issue in the policies as well as its implementation. And regarding automatic identification system, vessel monitoring system, both has been used, but they have not been implemented throughout the country. Also, the engagement of fishers in the policy planning and policy implementation has been very weak. And when it comes to a country's size like India, the implementation requires a lot of interstate collaboration, inter-agency collaboration, and this has been another weak point that we have found. Also, so far there has been no national plan of action that has been prepared by India as of now. So some of the recommendations that we give is to, first of all, strengthen the vessel tracking technologies, and that is first in its policy regulation as well as its implementation. And the second aspect is to have a more better coordination amongst the agencies who are involved in implementation of these policies, which has been pretty weak so far. And we also want to highlight the role of fisher communities in their decision-making process regarding the policies, because they are the biggest stakeholder in this. And for a country like India, I think there's need to be multi-layered strategy in order to address the diverse ecosystem or diverse marine challenges they are facing. So that's what we also highlight here. So yeah, for overall, we just want to highlight that this whole talk is about how the conservation issue in Diego Garcia or even in Chagos can be addressed, whether it is from the point of view of India as well as from Chagos inside as well. Thank you.
Thank you. Sorry for depressing you a bit, but one of the biggest threats to the Chagos marine life, and you know, with the future of the MPA a little bit unknown, we're worried for the future. So someone who shares these concerns about illegal fishing is Dr. Clare Embling, an associate professor of marine ecology at the University of Plymouth, who has over 20 years of experience researching marine mammals and knows only too well what the destruction of illegal fishing can do to marine life. She currently heads up a team carrying out the first dedicated study of cetaceans in the Chagos Archipelago to understand their distribution, their abundance, their habitat use and vocalizations, seeking to determine whether the Chagos Archipelago is one of the last tropical refuges for cetaceans. Is it?
Thank you. I look forward to talking about cetaceans because I think Tom came and gave a talk last year, didn't he? But I don't think there's that much known about cetaceans, and that's why we ended up doing this project. So I'm going to be talking about, I don't have an answer for this question quite yet, but is the Chagos Archipelago the last tropical refuge for cetaceans? And this is work funded by the Bertarelli Foundation, but some of the work was funded by the Garfield Weston Foundation. I wanted to put in this because this is not just my work, obviously. I put the ones in blue are the ones that are doing most of the work, which is the PhD students and the observers who are spending all of their time out there collecting the data. So that's our team. So why cetaceans? I mean, you might be surprised that so little has been done on cetaceans in the Chagos Archipelago, and it's always amazing to think that there's places in the world that have been so poorly understood. One of the main issues for me was thinking about it was in the Indian Ocean, one of the key threats is fisheries, okay? So especially the tuna fishery is the main threat to cetaceans in the Indian Ocean, and these figures are taken from some figures looking at bycatch. So the one on the top here is bycatch of baleen whales, so the big whales, and the one at the bottom is dolphins. And the graph on the bottom there is just how you can see that it's increased over time. So one of the guys who's collaborating on this project, Charles, he's been looking at this for quite a lot longer than I've been working on cetaceans. So not to say he's old. I hope he's not listening. I put my foot in it now. I think he's in Indonesia at the moment. But he did studies looking into the tuna drift, the tuna fishery, and that has massive amounts of bycatch of cetaceans, particularly small cetaceans, of about 100,000 individuals per year. And over the time period that he looked at, that's 4.1 million small cetaceans. This has likely reduced the small cetacean abundance to 13% of pre-fisheries levels, so this is a really, really big issue. Not only that, but cetaceans, especially the larger cetaceans, are prone to being hit by ships. And you can see on the bottom left here, that's where the shipping lanes are. And say, for example, around Sri Lanka, where my collaborator Asha DeVos works, then this is a big problem for whales there. So the Chagos Archipelago could be a refuge for some of these threats, okay? Because you can't fish there, or there's limited amounts of fishing there. And so it could be a refuge from all of this kind of like threats in the wider Indian Ocean. So what we're doing in the project, so that's a picture of Isha. So she's looking at the abundance, distribution and habitat of cetaceans from visual survey data. So we had Charmin Roof, who some of you may well know quite well, because she used to work at ZSL, and now Chris Robbins, who are out there collecting the data, and Isha's done quite a lot of the data collection herself as well. But we've also got data from the northern Maldives, so you can see right up in the northern atolls of the Maldives, working with Charles. So she's modelling abundance. So she's trying to calculate how many dolphins there actually are and how many whales there actually are, because we need to know that to be able to manage those populations, but also understanding where they are. And we use very standardised methods to be able to collect the data to be able to then estimate how many there are. So I'm going to be presenting some of her preliminary results. Then also we put a network of hydrophones around the Chagos Archipelago. Originally I had high ambitions to put them everywhere, and then I've lost a couple in deeper water, say over Sands Seamount. I lost 2 there. So we tend to try and go somewhere which is a little bit more accessible. So mostly shallow water. So you can see we've had hydrophones in Egmont Atoll. There was a month from Sands. Great Chagos, the south of Great Chagos Bank. We've got Middle Brother, Peros Banos. We've got one at Nelson that we hope to get back on the next one. And then we're going to be deploying in a couple of other locations as well. And Udayanga, who's based in Sri Lanka, he's analysing this data. He's analysing all the acoustics data. He's got data coming out of his ears. And he's looking at dolphin whistles. We found humpback whales, so I'm going to talk about the humpback whales. And he's also going to be looking at long-term data sets. So there's the CT-BTO data, which is... I didn't put that on there, did I? So it's for monitoring, actually, nuclear explosions. But it's very low frequency. It's continuously recording at 500 hertz, and we're able to pick up baleen whales on that. And he's got back 20+ years of data. So he's going to be able to look at long-term changes, and he hasn't quite decided whether we're going to look at minke whales or Brewer's whales quite yet. And then finally, I just want to mention some capacity building we've been doing within the Indian Ocean region, and I'll introduce that at the end. So Isha, sorry to throw up loads of pictures here, but Isha's first analysis has been doing is looking at the abundance and distribution of cetaceans in the northern Maldives atoll. Now, this was mainly because when we started the project, we didn't have much data yet for the Chagos, and Charles had lots for the northern areas of the Maldives, and it's a good place to compare against populations in Chagos as well. So what you can see here is a picture here of the distribution and abundance of just spinner dolphins. She's done all of the dolphins and whale species that there are found there, which is many species of cetaceans. So you can see that these are mostly island atoll associated, so mostly in shallow water. So I haven't got, you can see actually the numbers down on the bottom. So this is just for this area, so you can get an idea of the numbers of the animals that we're finding there. Showing the importance in all of them of sea surface temperature and driving the cetacean abundance. And that's what I think is going on in these pictures down here. So what you can see is you're thinking, oh my goodness, what's happening to the dolphins? It looks like they were quite high and they've dropped off quite dramatically. I don't think that's a change in the abundance of distributions. It's just movement patterns that are happening as a result of sea surface temperature. I mean, we don't know that for sure at the moment, but it seems to be linked to sea surface temperature. So animals might be distributing themselves differently within the atolls, or moving from other areas dependent on the environmental conditions, and this is important to understand. So what about the Chagos Archipelago? Well, we've got about 15 species of cetaceans recorded. Just to highlight a few, these are all of their Latin names, so picking out a few, I've highlighted the ones that are found kind of like more coastally. The most common ones are spinner dolphins and bottlenose dolphins. We get 2 different species of bottlenose dolphins. These are the main atoll-associated ones. So if you're seeing dolphins around atolls, it's most likely to be these 2 species. We do find them further away from the atolls, but they're mostly concentrated there. And we have had, and this is not a photo from Chagos, unfortunately. I haven't got a photo of the humpbacks from Chagos. I tried to choose somewhere fairly close. But we have had sightings of humpback whales in the Chagos Archipelago and calves as well. So bear that in mind when we talk about the acoustics. We've got very rare beaked whales. We've got quite a few different species of beaked whales. These are very deep diving species. we don't actually know how many there are in the world of many of these species because they're often very far from shore, but we do get ones between the Great Chagos Bank and the northern atolls, and between Great Chagos Bank and, say, like Diego Garcia. So we get beaked whales. We've had some longmans and a few other species. And then other species such as pilot whales and melon-headed whales. I've just shown you a couple of pictures there. And they tend to be found in those areas between Great Chagos Bank and the other islands. And then offshore, we're getting... You can see the green. We've had quite a lot of sperm whales found in the deeper waters, further away from shore, and blows of larger baleen whales as well. So you can see that they're distributed around the atolls. Now, bear in mind that this data is collected where the patrol vessel goes, because that's how we collect the data. It's not like we've got our own boat. So these are from the patrol vessel, so it's kind of biased to wherever the patrol vessel goes. So what Isha's going to be looking at next, she's just starting it now, is modelling those to look at abundance and habitat use, to try and understand how many we're getting in the Chagos Archipelago and what are the hotspot areas. So what about dolphin acoustics? So this is some of the preliminary results from, well, actually, it's just about to be submitted for publication. Firstly, Udianga looked at dolphin acoustics, and I'd collected data from a previous project at Egmont Atoll, so that's the data set he started with. And interestingly, although when I deployed it, I didn't see any, well, I saw one lone dolphin on Egmont looking a bit lost, and it did strand itself. It was a bottlenose dolphin. Other people have seen dolphins there quite frequently, but we put the hydrophone down, and what was really interesting is that nearly 80% of days we had dolphins, probably spinner dolphins, looking at their vocalizations, their whistles. But there's a real clear diurnal pattern. So like around dawn and dusk, you're getting the peak in the vocalization. So it's thought that they're probably traveling from offshore. They tend to feed offshore and then come into coastal areas or lagoons or whatever to rest. And that's what we think we're seeing there, is them traveling to and between the offshore waters and past Egmont Atoll. And I think it's quite amazing. This is 3 months of data, so some of the other sites have got longer amounts of data, but it's quite interesting that they're found there so much. This also matches up quite nicely with the work that Tom's done previously in Peros Banos, showing that spinner dolphins actually spend the day in the lagoon at Peros Banos resting. So that might be, they're not actually resting in Egmont Atoll, but they could be resting nearby. Yeah, and just to note is that of all the data that he's looked at so far, I've got another hard drive of data to send to Udianga that we haven't looked at yet. We've found dolphin whistles on all of them so far. What I found really exciting, and unfortunately, and I'm not going to impersonate a humpback whale, but there was a recording there, but it's not working, but that was humpback whales from the Chagos Archipelago. Something that I got really, really excited about was that we found humpback whale on all recordings except for Sand Seamount, but that's only 1 month of data. I suspect the timing wasn't quite right for that population, is we've got humpback whales on every single recording that we've got. So we've got non-song vocalized, so humpback whales could produce different types of vocalizations. The ones that you probably most well known, and the one I had there was a song, so the males singing to the females to try and to attract them. But we've also got non-song, so these are kind of like more social sounds or maybe feeding sounds, and we've got both of those, and we get the non-song only so far in the northwest monsoon, so that's kind of like December, January time, whereas we're getting song in the southeast monsoon, and we think that that's probably the population that's coming up from the south, okay? So that's really exciting, so that's what Udianga is currently analysing. Also, I've had a couple of dissertation students who've looked at the data, and there seems to be a clear diurnal variation in calls, so they're calling more at night, which is quite interesting. I don't know, it's dark. I'm assuming that they're just calling to each other because it's dark, but we don't know. But we think they might be different population of humpback whales. So what we're doing is we're sending around the vocalizations. You can see some of the vocalizations down there in the bottom here. We're sending them around to researchers around the Indian Ocean to find out whether they match up with their populations. Because they produce different calls, the different populations. So hopefully you can match them up and we can work out which population they belong to. Finally, I wanted to talk about capacity sharing. So a large part of the project is doing a lot of capacity sharing, or as Asher likes to call it, capacity No, she calls it capacity sharing. Everyone calls it capacity building. So we ran a training course in Sri Lanka in May 2023 to people, not only students, but people from local NGOs and government organisations doing statistical analysis and abundance estimation, bioacoustics. It was a 9-day, well, it's a 9-day course. And we also had a little mini conference. We're going to be doing the same in the Maldives this August, but focused mostly on statistics. But alongside this, we're working with two Maldivians to work with local school children. So Riaz has been doing weekends in he's actually the dean for the Maldives University, and in his weekends he volunteers going around schools trying to persuade people to study marine science. So he goes around to lots of little schools. There's a picture of him doing it down here. And then Hussein is going to be doing it with 16 to 18-year-olds because there's a gap there where they can't start working, but school finishes if they don't go on to A-levels at 16. So he's going to be doing he's done a weekend course on marine science that incorporates cetaceans as part of it. And then finally, we're putting together outreach in schools, teaching materials online. We're just starting that right now. This is focused on marine conservation broadly, so some of you might hear from me to get some materials for that. So it's going to be web-based resources for teachers that will be translated into multiple languages, so Creole, Maldivian, and the Sri Lankan languages. And hopefully it would be nice to share that around with some of the Chagosian communities as well. So that's it. So the question is the Chagos Archipelago the last tropical refuge for cetaceans? I'm not sure I can yet answer that, but what I can say is it's got a very high diversity of cetacean species. It has regular acoustic presence of the species. And it's a no-take marine protected area, so it's well protected from bycatch and any other human impacts. So that's a win for me. And the data that we are collecting will go into the management of the marine protected area, and also we're going to make sure it's disseminated to the wider Indian Ocean networks through the capacity building and sharing. And that's it.
Thank you. Thank you, Claire. That was great. Everyone needed to see some dolphins to lift our mood, and our adoration of the very photogenic dolphins, coconut crabs and seabirds. That's the next and final speaker, Jodey Peyton. She's challenging that. So Jodey is a research ecologist for 16 years who first visited Diego Garcia in 2018 for a Project Horizon scanning for invasive non-native species in the UK overseas territories. And she's got a background in both field botany in the UK and in the OTs. She's a regular contributor to Chagos News and a dedicated supporter of the Trust, which we're very grateful for. Jodey promotes use of open access tools such as iNaturalist to increase the number of biological records of species to help conservation managers understand the distribution of species around the world. In 2021, Jodey won a 3-year Darwin Plus project to update inventories of the terrestrial flora and fauna for Diego Garcia, and is here tonight to tell us 2 exciting important things. Firstly, there's a new Chagos species, and secondly, invertebrates are just as cool as sharks. Not sure, so tell us more.
Cool. Thank you, everyone. It's an amazing pleasure to be here with you. Sarah, you've done an amazing job organising it. It's just a privilege to hear so many fantastic stories. And yeah, Pete, you started it brilliantly, and it's just been, rising from there. So thank you for that. And so, my talk is obviously, they're probably not as cool as sharks, but hopefully I can share with you some of the cool things that we found during this project that we ran for a few years in Diego Garcia. And as you can see, like picking around in donkey poo, we're obviously not as glamorous as shark researchers either. But there's certainly a lot to see and a lot to find. And as Mark said, there's hopefully more that we don't know and we can look for. So as Sarah introduced, this project was looking at Diego Garcia and looking to see. In the 90s, ZSL did a really fantastic. ZSL have been the gatekeepers of fantastic data on invertebrates for the Chagos Archipelago, but some of this data was quite old, so we thought actually talking with the Bight administration, this is a really good opportunity to try and get some new data, basically, to see what we can learn a bit more about Diego Garcia for a start. And then also we were looking at updating the plant list, so we were working with Kew and Meise Botanic Garden in Belgium to update the plant list for Diego Garcia as well, and we're still doing it. Turns out it's quite a slow process, but it's fine. We'll report on that next time. But yes, we've been updating the plant lists and invertebrate lists using a variety of methods. We had fixed point sampling around the site, so we can go back to those, or others, we'll be able to go back to those in future. We were looking at sites across the island, so kind of the interest from this is invasive non-native species, and I'm sure everyone in the room knows what they are, but they're species that are introduced by humans outside of their natural ranges, and they have a negative impact on the environment or human health or the economy. So that was our kind of interest in ports of introduction. Is there any sort of differences in what we're finding? We're still kind of looking at that. So spoiler, we don't have the answers for that right yet. But anyway, we had a bit of a hiccup in Bahrain, and I think those of you who've been to Diego Garcia know that sometimes that happens. We had quite a long hiccup in Bahrain, but anyway, we did get there to Diego Garcia in the end, in June in 2022. And from our team, we had Rian Gwilym, Keith Ben-Season, Wolfgang Rebich, and Chris Malumphy, who's in the previous photo. That was from our team. And then we had Lamar Antipasado, who's a US Naval Facility natural historian. He's amazing. So we were all out looking with malaise traps, light traps, leaf sieving, suction sampling, all the fun things, looking for invertebrates. We found some really beautiful things. This fantastic trapjaw ants that you can see on the far side. They have these amazing mandibles, and that's obviously in a microscope. We had an event with with people on Diego Garcia, with the military and other personnel, to kind of show what we had found, and they were one of the highlights, I think. We found honeybees, as you'd expect, but these really cool ensign wasps. So these are cockroach parasites. So yeah, they will hunt them and predate and parasitize them. So they're very cool. We found lots of grasshoppers. We found 9 species that had been identified. There's an endemic species of cricket from the Chagos Archipelago, and that hasn't been recorded from Diego Garcia, but we found it for the first time, so that's really good. So that, you know, there's a lot to see, basically. There's this meadow cricket as well on the right-hand side. We found a giant Asian mantis that's introduced, obviously. It's the first record for Diego Garcia, I think probably for the archipelago, in fact, actually. Because it's got quite a sweet, I know it's introduced, probably having a problem, but it's got a lovely little face. Then it's going to get worse, people. I'm going to get more emotional. But yeah, so beetles. So we found this lovely transverse ladybirds. Some really cool rhinoceros beetles. Again, they're a problem for economic pests, but they're really cool things. And then just some chafers. But we've only been able to identify 9 species so far of the beetles. So what I should have said at the beginning is a lot of the challenges with invertebrates around the world, they're not very well studied. A lot's unknown about them because they're hard to identify. They're not always easy to find. There's a lot of effort that goes into kind of processing material from them. So yeah, it's not easy. We found some really nice flies. We identified 13 species. This one, the green one, it's a long-legged fly. Again, it's a parasitic species, so it'll hunt other species and they're just cool. Some very nice moths. That's very scientific, right? It's cool. They're cool. So moths and butterflies. We identified 18 species. This very nice heliotrope moth, which I think people who go there will see this a lot. It's very common. Found 11 species of centipede and millipede. Again, they are quite creepy crawly, but they're still very cool. Some really beautiful, really blue ones, amazing, really fabulous looking things. So that's through the leaf litter sieving, kind of doing things. So, a lot of traps, if you like trapping for moths, that's quite easy to do. They're kind of things we're drawn to them. But we were kind of trying to use as many methods as possible to look for invertebrates, see what we could, just get as much data as we physically could, basically. And we did. We got a lot of samples. There's a lot of sorting. So I spent many happy hours sorting through lots of samples, separating out all the different orders that we had, so kind of like whether they're beetles or, flies, whatever. 1,300 specimens were identified by the Natural History Museum and barcoded, so they're available on board, so they're DNA barcodes. The aim is to kind of try and help people going forward if they find something to try and make it easier to identify it going forward. But that was it was just an incredible process, really, just so exquisite. I know they're not sharks, but they are really cool. So these really cool web spinners, the ones in the middle there, I've never seen them before. Again, it's the tropics, they have amazing species, just really cool, really cool web spinners. Nice carabid beetles, so they're like predatory beetles. You've got these really beautiful blue little centipedes. The kind of the blurry one, the little tiny one, it's a fairy fly. That's actually a parasitic, it's one of the smallest insects. Fairy flies are some of the smallest insects in the world, certainly the smallest flying insects in the world. They're absolutely tiny, and they're beautiful. And just, to find that in a sample of, zooming in, what is this? And just finding this beautiful thing. And then the cool parasitic wasp at the end as well, with a crazy little waist. And this really nice giant water beetle. It was a really mesmerizing time going through the samples. Yes, exactly. Isn't it lovely? Thank you, Pete. Yes, he's been paid to say that. But I think it's really cool. I mean, unfortunately, it's a really bad invasive species in the sense that it has very negative impacts. But it's so fluffy. I mean, look at all that hair. But so this is a big-headed ant, Phaidalea megacephala. It is, unfortunately, a problem species. But it's one of the things we found. So Rian, she's from Gibraltar. She's an antex, a myrmecologist. She Pete has facilitated getting Rian some more samples of ants. But so Rian is a myrmecologist. She identified 25. She basically updated the list of species for the Chagos. So there's now 25 ant species. Some of them are quite problematic. In terms of the fire ants, there'll be a problem for people. We know this, but they're also a problem for things like turtles. And what we're particularly interested in is a species that hasn't arrived yet, as far as we know. It's Wasmannia arapunctata. It's a little fire ant. Any of you who's done any work in kind of the Americas or Hawaii, or you may have come across this, but it's an incredible species in terms of its impact. It can attack turtles, seabirds. It'll be a real problem species. And what we were worried about is wanting to find out what ant species are there, what species are there generally, because actually what both Mark's talking about in terms of sort of synergistic, kind of looking at this bigger picture. So ants, so some ants will just literally attack turtles, birds, they will actually have their own impact, but some also support and farm aphids and scale insects, and then they have they'll be then having a problem on the plant species, which then may affect the nesting birds as well. So what we were trying to do is get as much baseline data as possible about what is there, and so what kind of things in the future may be a problem for Diogasia as climate changes, as species are introduced, as the world changes and it gets more challenged, what we need to kind of prepare and help Diogasia prepare and the Chagos prepare for what comes next. What was really interesting, one other thing that's interesting about the work that we did, so Chris Malumphy, he works at FERA in York, so it's a institute where he's kind of looking at pest species. He said he found very few pest species, and that's, I guess, the history of Chagos is kind of protected in the way that other islands in the Indonesian haven't been. So actually he was very pleased to find so few, like scale insects and whitefly species, other kind of pest species that would have an impact or negative impact potentially on the plants going forward. And actually that's one thing that we were very happy to hear about. And I think it's again what makes Diogasia or the Chagos Archipelago very special. So it's just a quick run through of the different orders and the kind of families that we saw. It doesn't really mean anything other than the fact these are probably the sampling methods that we did, so we didn't really focus on the dragonflies and damselflies at Odonata. I know Pete's got a lot of work on that anyway, but we were kind of, the methods we were using were kind of selecting for those kind of different orders, basically. But it just shows there's a lot there. There's a lot of variety and there's a lot to see and there's still a lot to identify. We've got a lot of stuff in pots that need experts. It's very hard to find people who can actually identify these things. And that's why I wanted to do the barcoding, to try and help at least try and link some names to some barcodes. Another really exciting thing was that the surveys that we did found a new species to science. This Australopithecus chagosensis, I'm not sure how you say it, but it's a very cool little thing. Its closest relatives are in southern India, Sri Lanka, and the Maldives. I mean, it's probably the fact people just don't look for them. It's probably elsewhere as well, but first described from here, which is fantastic. And again, our surveys, there was only one known centipede species from the Chagos Archipelago before our visit, and we've added another 4 now, so that's, it's cool to add, to learn more as these kind of things are in the food chain and the kind of cycling of nutrients and things. And yes, as Sarah said, I'm really passionate about kind of other ways of sampling. So, going out in the field is very labour intensive. It's obviously very expensive and very difficult, but really, really rewarding. But there's things that can be done. Some species can be done, can be identified via photos, and that can be done with iNaturalist. When Q set up this project when they went out in 2018, and we were really keen to use it again to kind of spatially map where things were on Diego Garcia. And there's now almost 4,700 observations. There's almost 1,000 species. It's a really, if anyone's going out there and they're kind of bored of a day, if it's not quite perfect sea weather or whatever, or you're going to these outer islands, that would be amazing to get some more just records to see what is there, what we're looking at, because things like, the ants you couldn't do from iNaturalist, but it would be, there are other things that you could identify from iNaturalist, and they would be really useful to know about. And what Mark was saying about a guide maybe on the crabs, what we did for using this iNaturalist app, and I'm happy to talk to anyone about it if you want to later, or by email or anything. I'm very passionate about it. But you can make these guides, so they can actually be on your phone, you can download them, and so we made these for the project for us going out, and they need updating with the things that we found that are new, but they're all freely available. You can go onto the iNaturalist app, download it onto your phone, and if you see something in the field, you can then you can access it offline and just see what it might be if you're interested. As I was saying, this invertebrate work was part of a bigger piece of work, and one of those aspects, working with the Bioadministration and the GB Non-Native Species Secretariat, is around biosecurity. Because again, if you've got some tricky ant species in Diego Garcia, you don't want to be taking them out to any of the other islands. So kind of working with the kind of customs teams and with the Bioadministration to support, train the trainer of key things around biosecurity, especially around things like ants that are going to be that could be a massive problem. And really kindly with Pascaline Nellen and Lamar Antipasado, we managed to get these really lovely guides translated into Creole and Tagalog. So that was really nice, and they're available. They should be on DG somewhere, but hopefully with the Bioadministration team. And yeah, so basically, Pete managed to bring back some ant samples that have been collected this last year as well. And so what we're really looking out for is this little fire ant, because it's not on Diego Garcia yet. It's a very small, it's a really problematic species. It causes huge problems around the world in terms of also people being stung as well. And so basically if it does arrive, we really want to be able to act on it very quickly and knock it out before it spreads. And in terms of further work, we're waiting for the outcome of, it's a Darwin main, not strategic, that Pete was waiting for, and also this Darwin Plus local. We want to do more work on the spiders. We would like to do some red listing as well, for the Indian Ocean, and globally for the endemics, but maybe some other species of regional red lists. Basically working with the Bioadministration on a biosecurity manual, and also the US Naval Facility. We've got very good contacts there, so hoping to work together, work with more people to try and understand how we can all protect whatever happens in the future, make sure we can give it the best start now. And that's it. I just want to say, thank you to Sarah and the CCT team, but thank you to all the people who helped us, Rachel specifically, who got us out of many a pickle wherever you are. Yes, thank you. And my colleagues at CEH, where I used to work when I was doing this work, and of course the Darwin funding for funding it. Thank you.
Oh, thank you, Jodey, for such a wonderful talk to end our session. It's so easy to focus on what's perceived to be the cooler animals to attract an audience, but now we all think invertebrates are cool, although I think some scientists will challenge you on the ants. So thank you to all our speakers this evening. Can I ask everyone to give all 6 a massive round of applause? You've been brilliant. Now, I know that I've gone hideously over and everybody's hungry and I can smell that they're serving canapés. So I'm going to use presenter's privilege and encourage you to ask your questions in person outside at the reception rather than getting everyone on stage because we've gone quite late. And for those of you online, thank you for sending in your questions. I'll make sure I note them down and send them to the speakers and we'll get them answered. But before you go outside, if you just permit me, I just want to say a few tiny little things. There's still time to become a member tonight if you're not. Our chair, Chris Davis, over here, he will be at the reception and happy to sign you up, take your money, and you can also donate. So we do have a little gift for each of our speakers, which Chris will distribute, and an extra few for you, Mark. But just permit me to just personally thank a few more people before we go. Thanks to Chris, our chair, all the CCT board, many of whom are here tonight, for all their hard work during the year. Remember, they're not paid, so they give us all their time. And there's Bri and Nicole, especially with the internship, John Francois, who keeps me up to date on all Chagosian issues, and our long-suffering treasurer, James, because I'm not great with numbers. And thank you to everybody who's helped us. Also, thanks to Pascalina, who can't be with us. She updates the Chagosian community website. And Pete, our program manager. I'm going to embarrass you now, Pete. Now, everybody always says that people are replaceable. Just not true with Pete. CCT couldn't be without you, so thanks, Pete. And Martina, our incredible intern, our first intern. I couldn't have organised tonight without her helping me with the RSVPs and on all the social media. Sadly, she leaves us next month after being with us an academic year, and you're going to be missed enormously. So thank you for all your work this year. Thanks to the guys up there, the AV team. They stepped in last minute and they're just amazing. They've helped me with all the slides, Dave and Gareth. Thank you to the Chagosian community for coming tonight, communicating with us. I'm pretty sure it's hard to see this and not to be able to go home, so thank you so much, and let's keep working together. And finally, ZSL's Rachel Jones. I couldn't have done any of this without you, Rachel. You've been an enormous help to me in the 3 years I've been in this role. You're always ready to help, even though you're so busy. You've been a great friend to the Trust for decades. Everybody here on the front row appreciates it. So thank you, and thanks to ZSL for being a great host. So now let's go back to the foyer. There's food waiting and drink, and let's toast to the Chagos Conservation Trust and its work for the past and hopefully the next 33 years, whatever that looks like. Thank you all for coming.